Okay. Hi everyone, and welcome to our Market Garden webinar, a deep dive into soil health and soil testing. I'm Jacinta Lees and I'll be facilitating this webinar. Thank you for being curious to learn more about soil health and also taking the time out of your day to watch this webinar. I want to let you know that the webinar is being recorded, and please make sure that your microphones are off during each presentation. If you have any questions throughout the presentation, please pop them in the chat box and we'll answer them during our Q&A session at the end of our webinar. I also want to let you know that this project is being delivered by NRM South through support from the TasFarm Innovation Hub and funding from the Australian Government's Future Drought Fund. So I want to first start our webinar with an acknowledgement of country. So we pay respect to the traditional owners of Lutruwita, the Tasmanian Aboriginal people, and acknowledge their continued survival and connection with their land, sea and sky country that spans millennia. We acknowledge the many nations of Tasmanian Aboriginal people, past and present, as the traditional and ongoing owners of their respective countries within Lutruwita and the island. We pay respect to those who have passed and acknowledge today's Aboriginal people who are the custodians of this land. So today we'll cover a quick summary on the NRMs and our soil extension program. We'll have a soil monitoring observation presentation by Mitch at the Agrarian Kitchen. We'll have another soil monitoring experiences presentation by Stan at Backcat Farm. And we'll finish off with a soil health and test interpretation guidance by Declan at Region Soils. We'll have our opportunity for Q&A, and please let me know what you think of this webinar at the end when I'll send through a link. So NRM stands for Natural Resource Management. The NRM model has been operating in Australia for over 20 years. And today there are 54 NRM organisations nationwide. We have 3 in Tasmania. We have the NRM North. We have NRM North, Cradle Coast Authority and NRM South. So we all work closely and regularly with other NRMs. We collaborate on projects and share knowledge and expertise between our NRMs in Tasmania. We have similar values and goals. We focus on healthy landscapes and seascapes, protecting Tasmania's natural values and ensuring our actions have social, economic and environmental benefits. We also work together to develop our regional strategies. So while each strategy reflects the priorities of each region, which may be slightly different because of the different landscapes, species and industries to each sector, there's also a lot of priorities that are aligned across our state, like improving our soil health, having resilient landscapes, and also promoting best management practices. So our soil extension program aligns very closely with our regional strategy. This statewide soil extension program is delivered with NRM North and the Cradle Coast Authority. And within this program, we've been able to build capacity of land managers to improve soil health. We have been promoting sustainable, productive and profitable practices. We are wanting to build healthy and resilient systems. And we've been delivering this program through workshops, webinars, case studies, and our decision support tools that are on the slide at the bottom there. So now that I've finished off my little summary of NRMs and our soil extension program, I'm going to hand over to Mitch.
Can everybody hear me and see that okay? Okay. Thanks so much, Jacinta. Yeah, we're really stoked to be here, both on behalf of myself and the Agrarian Kitchen. I wanted to just give a little bit of context first before we sort of dove into what kind of soil monitoring we actually do. So if you haven't been to the Agrarian Kitchen before, it's a cooking school, restaurant, kiosk that's all attached to a garden. So the idea with our sort of roughly 1.3 acre walled garden, which is housed in the old Willow Court precinct in New Norfolk, in the old men's exercise yard. And the point of the garden is as a production garden to grow vegetables, fruit, and flower for the rest of the business. So we grow between 90 and 100% of the vegetables that the business uses. We also grow the vast majority of flowers that front of house do a really beautiful job of doing all our ornamental work across the business and decoration. And we sort of currently grow in between sort of 10 and 20% of the fruit for the business. The space isn't quite big enough to do, all the apples and pears and cherries and the rest. So we've just focused on things that we don't have access to, or things that are, going to be particularly delicious, grown in New Norfolk, and especially in a protected courtyard garden space. So that's what we grow. The other big part of what we do is exploring growing systems that overall ideally leave the landscape and the garden space in a better shape than when we found it. So we definitely subscribe to the idea of regenerative agriculture. We certainly wouldn't ever claim ourselves to be regenerative, but those goals are what we work towards. So yeah, we certainly favour systems that are organically based, but really we just work within our context. And so for us, of course, we're connected to the kitchen. So there's a lot of waste. So we focus on a lot of circular waste systems. So we collect all the food waste, all the, the meat, everything that's coming out of the restaurant, as well as bones and shells and paper waste, anything that's organic in nature. And we process that and make compost and other foliar sprays. And we're at a point now which has been really nice to reach, which we didn't actually expect, that we can drive all the fertility in our garden space just via these waste products. So we don't actually import any outside fertility. One other really key system in our garden is our sort of deep compost, no dig or no till bed system. I'm sure a lot of you are familiar with it. It's quite popular, particularly in the small scale market gardening space at the moment. I think it works really well on this scale. And it's how we set up all of our vegetable beds as well as our perennial beds as well. And one other sort of key system is this idea of lean systems. And really, in a nutshell, the idea is that we're as efficient as possible. Things are designed and planned to try and cut out waste as much as possible, both in terms of sort of labor and actual resource waste. And so really it's sort of a high intensity, small scale garden space. So what we're here for, though, soil monitoring. So what do we do to look at our soils throughout the year and over the years? So we use AgPath to test our soils. And we actually really only test our market garden soils. I guess a lot of the literature showcases that it's really annual vegetable production that has some, possible negative ramifications on ecosystems. And so for us, the market garden made sense as a spot to focus on soil health and monitoring soil over time. So the reason we chose AgPath was because they offer both chemistry tests. So we want to look at our chemistry, macro, micronutrients. We want to look at our carbon, organic matter as well. But we also want to look at biology, and we want to look at those things as connected to each other. So we send it off to her lab and we'll get the chemistry results back, but also biological analysis as well. We've also done a little bit of in-house quantitative microscopy work. One of our gardeners has done a bit of work with microscopes, but that's been relatively limited. So when we first started, of course, we wanted to get baseline tests, so we knew what we were starting with, and if we needed to make any big changes. So we took the first test in 2020, and what was surprising, what I had assumed anyway, was that there would be a few macro micronutrients that we might need to fiddle with. There might be a few things not quite right or in optimal ranges, but because it's been sitting there as pasture, you can see it's been mowed in that top corner. But it hadn't been running as an exercise yard for almost 3 decades, 4 decades. So I was just sitting as pasture. There was a big ash tree in there, and the council would just come in and mow it a couple of times each year. So yeah, our assumption was that the biology might be fine. It should be in good shape because there's been no intervention and no harassment. But we might need to amend. And what we actually found was the opposite. So the chemistry, in terms of for us for growing vegetables, was almost spot on. So there were a couple of things. Our zinc was a bit high. I think our selenium was a bit low. But overall, things were actually in really amazing ranges, which is a dream to really get started with. But the biology was a bit lacking. So our fungal bacterial ratios are a bit off. The diversity in the bacteria and the fungal overall populations seem to be lacking and were lower than what was expected. So ultimately, from that, the recommendation from Dr. Mary Cole at AgPath was that we really just need to stimulate biology. We didn't really need to add lots of different amendments to try and change any of the chemistry. It was really actually just getting our fungal, bacterial, protozoa, nematodes, all of this, soil life going to start cycling through, and combining that with heavy plantings of vegetables and other crops to really start getting some nutrient cycling going. So that's what we did. We applied fish, we applied molasses, we applied other biostimulants and other sprays, macro boost products, Nutrog products, some of our own products as well, compost extracts, other bits and pieces. And we adjusted the selenium. But because we do use this noding system, that's how we really prepare our space, and it serves lots of different functions. We did apply heavy applications of compost, and you can kind of see down the bottom there what that looks like. Since then, so the garden's now about 4 years old, but we've really only been producing properly for 2 years. So overall, we've done a soil test almost every season. So we've got 3 soil tests so far. And really, what we're trying to capture is any trends. We just want to see over time what's happening to our chemistry, what's happening to our carbon, what's happening to our biology. We know that, we're not doing any experimentation. We're not looking for anything causative, just looking for trends over time. And again, in the exploration of, say, regenerating a space, ideally we want to see those things improving, our chemistry getting better or not getting worse, our biology getting more diverse, populations getting better, that kind of thing. However, the results have been slightly different to what we expected. Of course, I don't have enough time to dive super deeply into our actual results. But here's just a sort of, little summary of what we've seen. And so basically over time, and this does make sense in terms of the heavy applications of compost, is our nutrients have been getting higher. And across the board, that's pretty much the case. And our biology, in some cases, has been getting lower, which, of course, is not what we're aiming to do. And so for us, that highlights a few interesting things and raises some interesting questions. Obviously, there's lots of different variables and lots of reasons why that could be happening. So we're cautious to be drawing, too many conclusions. But ultimately, it started to raise some questions about using heavy applications of compost too frequently. And so for us, I guess one of my assumptions as a gardener and going into this space was that, my focus on organic fertilization was that I might be able to sidestep some of the issues with soluble fertilizers. But of course, in compost as well, there are soluble fertilizers and soluble nutrients, I should say, as well. And so the hypothesis that we're working with that Dr. Mary Cole's kind of led is that our heavy applications of compost over time has, of course, increased the chemistry, which and has given plants a ready access to soluble nutrients, which means they're not interacting as much with the biology in the soil, which is then therefore decreasing the overall biology in the soil. And that's kind of the idea that we're working with at the moment. And so for us, how's it affecting us on the ground? What are we doing? So we had started to slow down compost applications anyway. So our first, port of call in terms of, our soil health is looking at our crops. And overall, our crops, are still productive. We measure, capture all our yields, and we compare them. And overall yields are great, and that's the look of the plants is fine. But we have seen some slight changes even over, the last 4 years. And so for us, we started questioning a few different things. Again, could be lots of different variables. New gardens, there's usually this little grace period. As pests and diseases get established, you can start to see some other problems and things crop up. But we'd started to perhaps use a little bit less compost anyway. But with this data, that's given us a bit more confidence to explore that route. So what we're doing now is we're using less compost, and as I mentioned before, we're making all of our own compost. We set the garden up using commercial compost, using Nick Hansen's compost, which has been great. But now we're making our own. A lot of it's coming from food waste, which again is higher in nutrients as well. So we've dropped that a fair bit. But so what we're also doing is focusing on how we can stimulate the biology, and trying to get their populations up and to get more nutrient cycling happening. Because overall, if there's lots of excess nutrients in the soil, we want to start cycling that so we can start using it. So we're looking at different, of course, sprays, so foliar programs. So we have a foliar program where we use fish that we make, fish hydrolysate from fish waste from the restaurant. We use whey from our cheese making in the restaurant. And we also use a few other products as well. And then we're also looking at other ways of stimulating biology. However, in the context for us, because again, no dig is not just about applying compost for nutrients. You're also applying these, deep mulch layers for weed suppression and for protecting your soil and a whole host of other things. So that also in turn has started to modify our weed management a little bit more. So we've had to bring back in a bit more occultation, which is the black silage tarp. We don't do a lot of solarization with the clear plastic. So to manage weeds a little bit better, because we're not having these thick mulch layers, we're using some more occultation again. And something else we're bringing in is more interplanting in effort to try and get more nutrient cycling, more photosynthesis from the plants going into the soil, and then really driving that chain as well. So going forward for us, we keep taking tests. Obviously, 3 tests is not a lot. The data will become more powerful as we actually get some time. There's going to be variables. There's going to be, it's really, it's not actually going to make, the trends aren't going to make a lot of sense until we actually capture a large range. So we'll keep doing that. We're also looking at getting other interpretations, and we might hear some of those today from other soil scientists and other academics, because there are different perspectives on these processes and different ways of working around them. So we're always eager to learn more from different perspectives. As I mentioned, using other methods of biostimulation, so using other mulches, using fresh organic waste, from the garden mulched up, applied directly to soil in small amounts to cycle microbial populations or get them to be breaking stuff down, and also tools to measure that. So we've been looking at the microbiometer, but trying to maybe measure microbial biomass or respiration as a tool as well, to understand what techniques we use actually directly impacts biological populations in the soil. And finally, we're also looking at doing some in-field sap analysis. And I'm sure as a lot of market gardeners out there, it's always hard because you grow so many different crops. So it's a bit cost prohibitive to be measuring all your different crops. But we're looking at just selecting a few different crops just to get, real-time readings of chemistry and other.
Seems like Mitch must have frozen there. Thank you for your presentation. That was amazing. Stan, would you like to share your screen and start your presentation?
Yep.
Thank you.
get that going. Okay, thanks Jacinta for organising it. Thanks to NRM South. We've been market gardening on our land since 2010, and over that time our practices have changed a lot, as we've learnt. We've always tried to make sure that whilst we grow, delicious vegetables, that we're looking after all of the biological systems on our land. Neither my wife Bryony or I are farmers, or we have no farmers in our family. We're both scientists, specifically molecular geneticists. For the last years of my career as a scientist, I ran the microbial ecology group at CSIRO in Hobart. I'm not a microbiologist, but it was the time when deep DNA sequencing and metagenomics were being used to describe microbial communities. Most of the vast numbers of microbial species in the environment can't be cultured or identified using traditional biochemical or microscopic methods. We would take entire environmental samples, water or soil or sediment, extract all the DNA, sequence it, and then reconstruct genomes computationally. Doing this, we could identify organisms and work out their relative abundances at a site. The reason I bring that up is because there were some key things I learned from my time doing this work that have influenced how I think about soils. So, as everyone knows, microbes cycle all the nutrients. And one really important thing, it's a fundamental of microbial ecology, is this concept of everything is everywhere. Every sample contains a large number, thousands to millions of different microbial species. A few of them dominate, but the vast majority are present in exceedingly rare quantities. But when environmental conditions change, some microbes, including ones that were rare, now thrive and multiply while others struggle. The composition of the community changes to adapt to the new conditions. A really great and extreme example of this is in sites where there's been spills of fuel or oil, for example. Microbes that can metabolize those hydrocarbons suddenly seem to appear out of nowhere. But it's not that they weren't there. They were there. They were just really rare. But now that conditions suit them, they are thriving. This is how diverse microbial communities as a whole are resilient to environmental change. So yes, microbial communities are self-organizing and they're driven by the environmental conditions present. And what we found and what a lot of researchers found is it's very difficult to change a community by adding microbes, but it's much easier to change the community by changing the conditions. So bearing these concepts in mind, this is what we try to do on our farm with our soils. We aim for minimal disturbance, which is mechanical disturbance such as tillage or biochemical disturbance. And what I mean by that is, inputs like fertilizers, not using soluble nutrients, not applying too much compost at once, and not using herbicides or pesticides. This lack of disturbance should allow soil structure to increase and therefore niches within soils to diversify. And this should also prevent the kinds of changes that can cause long-term disruption to community structure. We try to build soil carbon as this is microbial fuel, and we grow diverse plants as continuously as possible. Plants select microbes within their rhizosphere by feeding them species of specific cocktails of root exudates. So doing this should maximize microbial diversity. So how do we do this? Well, here's a aerial view of our farm in drought conditions. This was just this past summer. I don't know if you can see my mouse, but I'm going to just refer to a few things subsequently in this talk. A big proportion of our property has a wildlife paddock within which we graze, rotationally graze a couple of sheep, and we have free-ranging turkeys. This is what we call the middle block. That's our bigger area of production, and then up here is our top block, which we've been growing on for the longest. And there's a paddock out here which is open to wildlife, which is later on will be referred to as the east paddock, and then over here is the west paddock. So the blocks used, we do different growing systems on the two different blocks. The top block here we have set up in the last few years as a deep compost system, similar to what Mitch has described, and it's not tilled. The middle block is more traditional no-till, I guess. So we have slightly raised permanent beds that are occasionally power harrowed, not rotary hoed, just power harrowed, and with a light composting a few millimeters on the surface prior to planting. We amend the beds prior to harrowing or into dibbed holes with our organic fertilizer based on soil tests, which I'll cover later. All our plants, or most of our plants, are seedlings that are planted by dibbing, and we use our old paper pot system with a homemade 4-pronged dibber. I'll just show you that. Sorry, I should have this slide here. And we also do a lot of cover cropping. So we plant cover crops with an earthway seeder, so we can do it in straight line, so that when we remove the plants, we can leave the roots in place. And we don't slash and incorporate mechanically our cover crops anymore. We usually just leave them on the surface and cover with the tarp. We also make compost on site using our homemade, using our home poultry manure, plant residues, biochar that we produce on site, and grass or hay. This is just a picture of the dibber that we use to plant. And using the paper pot system, we don't use the transplanter anymore. We just unzip seedlings like this. And then they're quite quick to plant. Not as quick as the paper pot transplanter, but pretty quick. On this bed, we're actually planting through grass mulch as well. So I guess the upshot is that we are trying our best not to disturb the soil, as I said before. We were growing fine. We were growing great vegetables for a while, and then in 2020 we had a bit of a problem. Some of our plants didn't look that great, particularly our spinach looked a bit yellow. So we got our soil tested at that time and did some sap analysis as well, and we used Garth Coughlin as our agronomist, and Agvita did the testing. You know, I should have thought about this before, because, you know, although we don't need to add microbes, we need to supply the minerals that microbes and plants require. And because we're exporting a lot of plant material off our land when we sell our vegetables, so we need to supplement that, which I thought we were doing fine by just using the original cough recipe from Steve Solomon and small amounts of compost. But the soil testing showed that which we did in 2020, that showed that we were severely potassium deficient and mildly copper and boron deficient, as well as being a bit high in pH and magnesium. So we changed our amendment regime based on what Garth suggested that we do, and we tested again this year in October, with thanks to Jacinta and NRM for helping out with the sampling. We also took a couple of samples from the paddocks to see what our non-market garden soils look like. Okay, I'm going to show a set of graphs now, and I'll just talk through this first one a bit more so you can understand what the colors and stuff mean. So blue bars are the values from 2020 and green bars are the values from 2024. In the east and west paddock, we only have 2024 because we didn't do that baseline sample in 2020, unfortunately. As you can see, our pH is, you know, pretty much neutral, just a little bit high in our top block, pretty much bang on 7 in the middle block, and quite acidic in our unamended paddocks. Our organic matter is high, but it's interesting to us looking at these numbers because the paddocks are high, as you'd probably expect for perennial pasture. Our top block, which is where we've been growing the longest, is lower than our middle block, but it seems that our organic matter, even though it's considered high, it is dropping where we grow vegetables. And I guess it's just the nature of growing annual vegetables. Even if you do no-till and add organic matter and have plants growing all the time, it's still not the same as growing perennial pasture. It's a shame we can't compare this to what would have happened if we'd not cared about our soil, I guess. Magnesium is pretty high, which is, it's pretty close to within the limits. In the top block, it's a bit too high, and we suspect that there are a couple of reasons for this. We'd been using the original Steve Solomon cough recipe, which has dolomite in it, and we stopped using that once we saw our magnesium was a bit high, but it's still gone up in the top block, and this is most likely from the deep compost that we added into top block since 2020. I guess there's quite a bit of magnesium in the compost that we use. We also use the Nick Hansen Cherries Tasmania compost. And as you can see in the middle block where we've stopped using dolomite, our magnesium levels are dropping. We are very high for zinc in both our vegetable growing areas. And I guess I'm not sure what the source of it is, but Declan might have more to say about zinc. It's potentially come in our compost or whatever other amendments that we have. It doesn't seem to be showing any signs of toxicity, so we'll just have to keep monitoring to see what happens to zinc. So potassium, which was the thing that we were most deficient for, which is what caused us to actually seek out soil testing. You can see we were really desperately low for potassium in both the top and middle block in 2020. And we've been supplementing with potash sulfate since then, and the top block seems to have gone up, but strangely, we've had no change in the potassium levels in the middle block, although we haven't had any of the growth issues that we saw in 2020. So that's an interesting one. We are deficient in boron, but with supplementation, we are able to at least keep it consistent, but potentially we need to increase our boron supplementation. And similarly for copper, we're on the low end for copper, but maybe it's not drastically low. And I think the last micronutrient, manganese, we're a little bit low for, and maybe the values are going down. We were recommended not to add manganese to the soil because our soils are pretty high in iron, and manganese competes with iron, so adding it to the soil wouldn't have been effective. But from just now, from this year, we are doing a little bit of foliar manganese application to try and correct some of the manganese potential deficiency. And actually, we did do a foliar application of chelated manganese on our garlic, and although I don't have a proper control, it seemed to have a dramatic effect on the look of the garlic. So it's interesting. With potentially there has been manganese issues that we haven't really known what to look for. So based on the 2024 soil testing, this is our current amendment regime. We are no longer adding any ag lime or dolomite until the pH approaches 6, or maybe not quite 6, but we've got plenty of calcium and plenty of magnesium in our soil. So the pH is a bit high, so we'll leave those amendments out. We use canola meal as our nitrogen source. We were using blood and bone as well, but our phosphorus is actually pretty high, so we decided to leave out blood and bone and just use canola meal. And then supplement with potash, sulfate, boron, and copper at these rates that you see, and then manganese as a foliar application. So I guess for us, we probably will now try and do soil sampling every year for another 4 years, just to see how things go, and hopefully we can improve some of these deficiencies. Will be interesting to see what Declan has to say about what we should do or what we should try. So thanks, everybody.
Oh, lovely photo. Thank you, Stan. That was a very good presentation. And yeah, it'll be very interesting to see what your soil test results will be in 2 years' time from now. Declan, it is your turn for the presentation. Would you like to share your slide?
Thanks, Jacinta. Will do.
Thank you.
Now, which screen can you see, Jacinta? Can you see the...
I can currently see the presentation screen.
with the other slides lined up, or just the first slide?
I can see the first slide, but it's from a presenter's point of view, not from...
Okay.
Yeah, perfect. Yeah.
Okay. Right. All right. Thanks for your patience there. Right. Yeah, Declan MacDonald is my name. I'm the principal with Regen Soils. This is my own business, which I set up about 4 years ago, having done my time, as they say, with a few state governments, New South Wales, Victoria, and Tasmania. So it was nice to be back in Tassie a few weeks ago with Jacinta, and nice to be talking to a Tassie audience today. So deep dive is what I was asked to do. So I've got quite a bit to cover. So I'm going to go through the first part a little bit quickly, because I want to spend a little bit more time towards the back end of my presentation. So there's a lot of talk about regenerative farming at the moment. And the way I look at that is I ask, most farmers will say that they want to leave the land in as good or better condition, but there's not really a clear understanding of what that means. It might mean better fencing, water laneways, or better soil. I think it means better soil. And better soil means having, improving organic matter and biological function. Two things that really have been largely ignored, I guess, by modern day agriculture. There are these principles of regenerative farming which have developed over the last few years, and I'm sure you're familiar with these. Keep soil covered, maintain living roots year-round, minimize soil disturbance, integrate livestock if possible, not really in a market garden situation, and maximize crop diversity. And when we think about what are these principles trying to achieve, they're recognizing that the soil is a living system, and we recognize the soil is probably the most complex ecosystem on the planet. And when we get that into perspective, it changes the way we think about our soil. So central to this soil as a living realm is food, is energy. And that energy comes from organic matter. And the principal functions of soil, chemical, physical, and biological, are inextricably linked through organic matter. And organic matter punches way above its weight and has a really strong influence on each of these functions. One of the issues with conventional agriculture is we've taken a very chemical focused approach to food production in particular, and that has meant the neglect really of the physical and certainly the biological functions as well. And we call this sometimes the three-legged stool, and the three legs of a stool have to be in equal proportion so that the seat of the stool is functional. And so chemical functions are extremely important in soil, but just as important as physical and biological functions. It is a system, and we need to work it as in harmony. So let's talk a little bit more about organic matter. So these are some of the things I want to cover off on today. Understanding organic matter, Why is soil disturbance an issue? What happens to soil organic matter when we disturb the soil? What should I do if I need to disturb the soil? How much organic matter does my soil need, and how often does it need it? So we'll step through some of these questions as we go. So starting with understanding soil organic matter. Well, it is the component of soil that consists of plant and animal detritus at various stages of decomposition, and I've underlined that for a reason, and I will return to this, but I want you to bear that in mind. It's made up also of cells and tissues of soil microbes, very important, and substances that soil microbes synthesize. Simple definition from Wikipedia, but I'm happy with it. So is all soil organic matter the same? Well, typically we organize our thinking around soil organic matter into three pools. Nature does not organize soil organic matter into three pools, but for our purposes, it helps us to think about it and therefore to manage it in this way. And really the difference between the pools relate to the speed at which these organic components turn over in the soil. So when we talk about turning over, we're talking about decomposing. So labile organic matter decomposes really relatively quickly. This says 1 to 5 years and 20 to 40 years, etc. But labile organic carbon can turn over in days or weeks. You know, if you pick a clover leaf and drop it on the ground, is it there tomorrow? Probably not. Even if you mark the spot, it might be gone. So particulate organic carbon is essentially that material that's very fresh, that's where the energy in it is very available, and it gets attacked and invaded really quickly by macro and micro organisms to consume it as an energy source. As organic matter is processed more and more by a range of different organisms, it gets converted into much more complex molecules. And we use this term humus to describe these complex molecules. Now, nobody has managed to say that's a humus molecule, because humus molecules are made up of a multitude of different molecules that combine together, that are loosely attracted to each other. But in this highly complex form, these organic carbon molecules take on a range of properties which are highly beneficial for soil function. Some of those properties are water holding, and some of those properties are nutrient holding. But of course, as generally the largest pool in the soil, it is a really important pool for energy, for microbial as microbial food. And then we have resistant organic carbon, protected humus charcoal. It's nearly always derived from fire. This is the recalcitrant carbon, and it's what we're doing now with our current interest in biochar, putting that into the soil because of its stability. But its stability means that it's not really much of a food for microbes, because they blunt their teeth on it. They can't get at it. It's so stable. It's very resistant to microbial attack, which means it hangs around for a while, but offers up other functions in the soil, such as water, nutrient holding, and importantly, habitat for microbes. So this is a neat little graphic here of, that continuum. So we have, fresh organic material on the left-hand side, and as we progress through to darker material that becomes more soil-like, we're progressing through from that particulate organic matter through to humus type organic matter. And along the way, carbon dioxide is evolving out of this as a product of microbial respiration, which is part of the reason why we have a concentration of carbon in these compounds. So soil stores carbon by essentially this very simple diagram. Carbon dioxide is taken from the air into a plant through photosynthesis. It's converted into sugars, which are carbon-based sugars. They help to the plant to form secondary and tertiary structures, but also some of the food that the plant manufactures then is exuded through root systems to in turn put carbon directly into the soil and into the soil food web. I liken this process to the economy. Now, I am no economist, and economists will probably shoot me for what I'm about to say, but my very kind of simple understanding of the economy is, when the economy is going well, everybody's spending like drunken sailors. When the economy is going badly, people stop spending. So the money doesn't evaporate necessarily, it just stops moving. So in the soil, because carbon is the primary energy source, is really the currency in soil, it's really important that it moves, and it moves freely. So when we talk about nutrient cycling, we're really talking about organic matter cycling, because organic matter is a huge reserve of nutrients in the soil. So when we want nutrients to cycle, they're packaged in organic matter, and we want that organic matter to decompose and to release the nutrients that are in there. And the more that that happens in a healthy soil, the more nutrient is available via microbial symbioses to the plant, and the better our production is, and the lower the reliance we have on external inputs. So if we look at this through the lens of a market gardener, if you're starting off and the farm has been run hard or run conventionally, I always say don't make any sudden moves if you want to farm organically or regeneratively. And we talk about, periods of between 3 and 10 years for a soil to come good, whatever that means. So the reason for that is that sandy soils generally respond quickly. They're your maybe 3-year to 5-year response times, and clay soils twice as long as that. But we say, what is responding? Typically, in a market garden situation, we will add a lot of organic matter into that system, but the real question for us is, can the soil digest it? So we can take a farm that's been run conventionally, that doesn't have a lot of biological activity, and we can dump a whole heap of organic matter in there, but we might just give the soil a big dose of indigestion, because it's not going to start turning over straight away. And we might say, oh, I put on all this compost and I'm a bit disappointed with the result, because, I didn't see a big response from it. We need a response because it's the microbial activity that's going to give us a response, and it's going to take time to build up those populations. So certainly adding compost is a very good first and necessary step. So we also need to understand how plants grow. And this neat little graphic here is showing a conceptual colonization of a plant root, which we can just see the tip of a root here on the left-hand side of the image. And the illuminated parts are a conceptual idea of mycorrhizal fungi. And the beauty of mycorrhizal fungi and the importance of plant-fungal relationships is that fungal hyphae are far, far smaller in diameter than the smallest plant root hair. And so they can get into spaces where plant roots can't access, and in so doing access water and nutrients, and in turn make them available to plants. So this is the way that nature has evolved over millions and millions of years. A central part of this is organic nitrogen. So nitrogen is a really important part of plant production. And Gary Zimmer, who's an American dairy farmer, organic dairy farmer, has this expression which I really like, which he says, you've got to earn the right to use nitrogen, which I didn't understand for a while until I figured out, I think what he's talking about is there's no point just throwing nitrogen onto a half-dead soil to make your plants grow. It will work if you're using something like urea, because urea works very well on half-dead soils. But if you want to optimize your nutrient use efficiency, you've really got to make sure that the soil is optimized to be able to accept and use whatever nitrogen we might choose to apply, if we choose to apply it. But what this graphic is showing here is that organic nitrogen, which is bound up in the soil's organic matter, is by far the biggest pool of nitrogen in the soil. And when we start adding nitrogen fertilizer, it might only be a relatively small percentage, but because it's in such availability and such soluble form, we're messing around with a core ratio in the soil, which is the soil's carbon to nitrogen ratio. And when we start tipping it below about 12:1, we're tipping it in favor of microbes, and that enables the microbes to attack the soil organic matter at a rate higher than it should, which is fine if that's what we want to happen, but it's a completely unsustainable situation, because eventually the organic matter will be run down. Similarly, another beautiful mechanism for nitrogen activation in the soil is found in relationships between nitrogen-fixing plants and, say, cereal plants. So a grass and a legume were always, were made in heaven together. Nitrogen will be transferred between the two. The grass plant will stimulate mycorrhizal germination and growth, and it's the exudates from the plants that will stimulate mycorrhizal start growing. They in turn will colonize the bean plant and will facilitate nutrients transfer between the two. So it's a very elegant system that this is an example of. So this is how plants grow and how soil works. So our role really in a regenerative context is to mimic this as much as possible and not do anything that's going to kind of stuff up these delicate and sophisticated symbioses that have developed over very, very long periods of time. But then along came Jones with all our wonderful tools, and we started feeding the plants, and feeding the plants made the friends redundant. So the plants stopped feeding its symbiotic partners and became dependent on these external inputs. So we need to understand why soil disturbance then is an issue. One of the principles of regenerative agriculture is minimize soil disturbance. Now, it doesn't say eliminate soil disturbance, it says minimize it. And minimizing it means only do it if you know what you're doing and you know why you're doing it. A really important thing to understand is that the spatial distribution of soil microbes is not uniform. And here we have two conceptual soils, which you might recognize. This one here is really quite a fertile soil, and as a result, it is sustaining all these spots of macrobial activity, if I can call it that. And you can see that there is really quite high populations in this soil. And in this part of the soil in particular, gee, it must be more fertile than the soil over on this side because it's sustaining a much higher population. If we look at the soil on the right-hand side, we'll see that the macrobial populations are scattered around the edges here, but there's very little in the interior. But the point in both of them is, in spite of the relative fertility of these two lands, the populations are still not uniform and thoroughly distributed. Now, this is very important to understand in soil, because when we start disturbing soil, essentially we start homogenizing these environments. So to come back to one of my original points, to enhance fertility, soil organic matter must decompose. And this is from Jethro Tull, and anybody who is a 70s child might remember this group that took their name from this guy who wrote about one of the earlier writers with the advent of the printing press about farming practices. And he observed that tillage enhanced decomposition of soil organic matter, which in turn promoted plant growth and fertility. And the lesson here was observed from where pigs uprooted the soil. They saw that plants grew much better after that. And yes, it might have something to do with the manure that the pigs left behind, but it has more to do with the fact that the pigs have mixed up the soil with microbially separated communities and basically mixed the microbes in the soil together. So we had a boom in microbial activity. But Jansen here in 2006 said soil organic matter is far more than a potential tank for impounding excess CO2. It's a relentless flow of carbon atoms through a myriad of streams, some fast, some slow, particulate organic carbon cycling, humus organic matter cycling, wending our way through the ecosystem, driving biotic processes along the way. So we want to manage this decomposition cycle. So he also says, in our newfound zeal for locking up carbon, do we overlook a fundamental precept that organic matter has most benefit biologically when it decays? So if I need to disturb the soil, well, firstly, I do want to have a focus on keeping soil covered. I always say soils weren't meant to see the sun. So to the greatest extent possible, we want to keep the soil covered. We want to apply corrections as required. And if we're disturbing the soil, that is the time to get lime into the soil, because lime is so insoluble, it really needs to be mixed in. Gypsum's a bit more soluble, and we really want to get compost into the soil too, although surface applications are acceptable. We want to include a rotation crop with soil organic carbon sequestration as a goal if we can, and that might be a green manure crop. And we might want to apply supplementary inputs to promote fungal growth, because bear in mind, when we disturb the soil, it's the fungal community that we impact most severely, because we basically smash up the fungal hypho networks. And bacteria will respond very quickly post-disturbance, but fungi take longer. So things that are going to help feed the fungus, such as humates, fish, or mature compost, are appropriate. And of course, living roots year-round to the greatest extent possible. When we add compost, we're adding fresh residues, and from the first days that we're adding compost to months to years to decades, we're moving through this cycle where carbon dioxide is getting released at all stages through that. The process is intimately involved with soil organism biomass and metabolism. Some of that organic matter is going to be protected in aggregates, which is why clay soils hold organic matter much better than sandy soils do. And then they become fixed to soil particles, stable organic matter, or in some cases, we talk about mineral-associated organic matter. A few years ago, I developed this calculator because farmers were saying, okay, well, if I use compost, by how much can I reduce my reliance on fertilizers? So in this first part of the calculator, we dropped in results from a soil test and determined what the soil analysis was and what the levels of nitrogen, phosphorus, etc. was in milligrams per kilogram. Then we said, okay, we're going to add compost at 5 tons to the hectare with a 25% moisture content, giving us an equivalent of 3.7 tons per hectare dry matter application. This is the analysis of the compost, but this is the kind of the brains of this little tool here. Available nutrients from the compost vary depending on the element. So nitrogen in year one, maybe only 10% of it is going to be available. But in phosphorus, with phosphorus, we might have 40% available in year one. In potassium, we might have 90% available in year one. So we have all these different availabilities. And then in year two, that tends to drop down again. And year three, it drops down again. But bear in mind, this means that year three is still delivering something from year one's compost application. And that's what this dizzyingly interesting graph is showing, that when I add compost in year one, I get 10% of the nitrogen out. When I add it in year two, I get 10% from the fresh addition plus 4 or 5% from year one. When I add it again in year three, I get 10% plus 5% plus 2%. When I add it in year four, I get 10% plus 5% plus 2% plus 2%, etc. So it's in this way I have a gradually increasing bank of nitrogen as time goes on, where the soil becomes self-sustaining in nitrogen supply. This is why it takes 3 to 5 to 10 years for a soil to come good, because it takes that long for the microbes to be able to be turning the organic matter over at a sufficient rate to start delivering nutrients to a crop at a level that sustains a profitable crop production. So when we talk about how much organic matter does my soil need, this little graph here gives us some understanding of the importance of how we apply organic matter and when. And this is coming from an experiment that comes from Rothamsted in England that started in 1852 and is continuing to this day. And there's essentially 3 treatments: control, where there was no additions. And that's really just sitting there at a fairly stable rate. There might be a slight decline, but basically it's not changing. The manure addition was started and stopped after about 15 years. And then we had this slow decline in organic matter levels over time back to this point of equilibrium. And in the third example, we had manure addition maintained. And as you can see, the rise was sharpest at the front, but it has continued to grow. And what this has shown is that soil carbon changes take place over long periods of time, but it also tells us that soil carbon storage capacity is finite for a defined range of input, and the largest changes happen early. It's also finite depending on your clay content, and that's a really important point. So a sandy soil is not going to be able to hold nearly as much organic matter as a soil with a higher clay content. And this importantly also shows that management changes that build soil carbon must be maintained to ensure soil carbon continues to increase. So when we talk about how does that translate to how much soil organic matter my soil might need, this is an example from cropping soils in New South Wales, but it serves to illustrate a point that when the land was first cleared of native vegetation and the land is converted to agriculture, we have this precipitous drop in soil organic matter. And then we get down to this level here, which is often referred to as the equilibrium level. This is a level at which soils are able to protect organic matter from further decay, but this can get very low in many soils to a point where that soil struggles to function and therefore becomes entirely dependent on external inputs. This is showing us that although we may never get back up here to the original level without putting it all back into native vegetation, we have considerable potential to increase soil organic matter. So this graph is showing a drop of soil organic carbon from about 70 tonnes per hectare down to close to 35 tonnes per hectare. In very round figures, we can call that 7% soil organic carbon drop down to 4% carbon. So if we say, okay, we want to increase it by 2% organic matter, we have a rule of thumb that says to replace 1% soil organic carbon requires 50 plus tons of organic matter per hectare. That is a lot. And if you're going to provide that with compost, that's an expensive proposition. In a small scale, that might be feasible. But again, we don't want to turn around and dump 50 tons of organic matter on per hectare in one go because of that digestion, that capacity of the soil to digest that material and all the nutrients within it in one go. So like I said at the start, we don't want too many sudden movements when it comes to changing soil management. We want to take the soil with us and lead the microbes through to higher populations. So too much of a good thing is too much, and that applies to organic matter as well as synthetic fertilizers. We can overfeed a soil. Soils with excessive compost applications can develop very high concentrations of nutrients such as ammonium, calcium, magnesium, potassium, and sodium. Too much of any nutrient can inhibit the uptake of other nutrients, resulting in deficiencies. Composted manure is generally higher in salts than composted vegetative matter, and in tunnels in particular, soluble salts can accumulate to excessive levels because leaching is interrupted by the tunnel, basically. Raw manure can be very high in salts and ammonium and is not recommended for use in tunnels. And most importantly, the surplus of organic food can also make the friends redundant. So even though, as we've talked about, the solubility of things like nitrogen and phosphorus and potassium is governed, the release of those nutrients is governed out of organic matter, out of compost. If we're putting on 50 tons, those small proportions are suddenly going to mean that we're swimming in a pool of soluble nutrients, exactly the same way as if we dump on a heap of urea or superphosphate. So too much of a good thing is still too much. So when we ask ourselves the question, how often does my soil need organic matter? We need to understand, firstly, is all organic carbon the same? So as we said before, it helps us to think in the context of three pools of carbon in the soil: particulate, labile, and active. They're all words for the fast pool. Humus and passive are words for that kind of slower pool, and recalcitrant, resistant, inert, or char, they're all words used to describe that very slow pool. And that very slow pool, is brought to prominence in recent decades by the rediscovery of the terra preta soils in South America, which you might be aware of, and that was a product of indigenous agriculture. So in terms of the residence times of each of these pools, the particulates are weeks to 1 year or maybe more, humus years to decades to centuries, and resistant can be decades to centuries, even to millennia. So what makes the difference in residence times is the chemical makeup, the level and the type of biological activity, and the location in the soil. Now, this was some work done by CSIRO near Harden in New South Wales, and they looked at 7 different soils, and they looked at the organic matter composition of each of these 7 soils. And whilst these first 3 all had fairly similar levels of organic matter, you can see that this first soil here, soil 1, had a small proportion of particulate carbon, a large proportion of humus carbon, and a large proportion of resistant carbon. This one here, large proportion of particulate, very large proportion of humus, not much resistant, etc. This one, very large proportion of particulate carbon, not so much humus, and not so much resistant, etc. So what kind of soil have you got? What are the proportions of your organic matter, or what are the implications for your management in terms of how much of these different pools you're likely to have? So if you're growing green manure crops, there's a reasonable chance your soils are going to look more like soil 3 or soil 2, because if you turn in or even tarp a green manure crop, we're putting a lot of particulate organic matter into that system. So we're going to put a lot of very readily available organic matter into that system. It depends what you're looking for, therefore. So if we're looking for healthy, productive soils, we want to have high soil carbon and fast turnover. And we want fast turnover to release nutrients, to drive plant productivity, to drive an active microbial population. And that's probably going to be more bacterially dominated. And that's where soil number 3 would function best, probably in a market garden situation. If we're looking to offset soil carbons for trading or long-term sequestration, we want a high soil carbon, of course, but we prefer a stable, long-term, slower turnover with lower microbial action that's probably going to be more fungal dominated, with lower nutrient release rates. And lower does not mean bad, it just means it's not a vegetable growing soil where we want higher levels of nutrient transfer. And so soil number 1 is what we would be looking for there in terms of the kind of organic inputs that we're adding to that soil. And management very much influences the proportions of soil organic matter. So again, this was some work from CSIRO, and in these 3 soils, the virgin soil had 118 tons of particulate, the labile organic matter. The pasture, very similar, had 116 tons, but look what happens in a cropping situation. A small amount of disturbance and the particulate organic material gets munched up really quickly. So a very, very small pool. So this soil is definitely going to be relying on fertilizers to grow the crop, because there's not a whole lot of biological activity that's cycling nutrients anyway quickly enough to support an annual crop like wheat or canola. So the conventional approach to soil organic carbon sequestration is to grow more. Grow a bigger crop, put fertilizer on, grow a big crop, that's going to increase soil organic carbon. But that has not turned out to be true, because when we add fertilizer, we tilt that carbon to nitrogen ratio in the soil, and we basically go 3 steps forward and 2.5 steps back, or even 3 steps back. New research is showing the potential importance of microbial-derived soil organic carbon. And what this work has shown is that soil organic matter with markers of microbe-derived lipids, proteins, and chitins, in other words, the compounds that you don't find in plant material, but you will only find in the likes of fungi and bacteria, in this particular study, 34% of the soil organic matter relative abundance came from microbes. And in this particular study by Kuhnen et al., this was a CSIRO study from 2020, microbial detritus contributed 59 to 64% of total soil organic carbon. And fungal abundance is particularly important for carbon sequestration. So in this diagram here from another research paper, it's illustrating that the microbial biomass might be less than 5% of the soil organic matter, but the microbial residues and necromass, so the mass of the dead bodies of microbes, is up to 50% of soil organic matter. So it's a really significant contributor to soil organic matter. Now, I'm just going to flip over quickly and talk about soil testing in this context. So my rhetorical question for you all today is, what's the secret to healthy soil? And the quick answer is air. Not nutrients, not organic matter, not mineral particles, because without air, this is not going to survive and thrive. This being the organic matter component, both the plant roots and the like, but also all the organisms that are working with this organic matter. I always say we can live for 40 days without food, 4 days without water, and 4 minutes without air. Most of what's in the soil is aerobic, so it is critically dependent on air. So soil structure is essentially the soil's aeration and drainage network, and it's critically important to microbial function. So when we talk about soil structure, what does it look like? Well, it looks something like this. Lots of marbles, lots of tiny marbles in a jar that have lots of spaces in between for root exploration, for microbial habitat, but most importantly for air and water movement. If you have a cloddy soil like the soil on the right-hand side here, we have roots that are growing around these big clods because they're too dense to grow into them, and roots are going to be struggling to form associations. That soil is going to support a much lower microbial population and lower cycling of organic matter. This little graphic here just shows what happens when we compress the upper level of the soil and we flatten it out and we close off these spaces and we impact on the soil's ability to breathe and drain. Now, the things that impact the soil's structural integrity are principally organic matter. It does the heavy lifting in terms of maintaining the pore structure in soil that facilitates that aeration and drainage. And calcium is the next most important, and magnesium also plays a positive role, providing it is in appropriate measure. So when we talk about the ideal soil, we want our soil to be calcium dominated, somewhere between 57 and 78%, depending really on whether we have a sandy soil, 57%, a clayey soil, on the upper end of that. We want sodium to below 5%, magnesium 12 to 25. And if these are out of whack, if we've too much sodium, if we've too much magnesium, it means we've got too little calcium, and we've got these are very weak agents when it comes to supporting structural integrity, and that's where we have a collapse of our aeration and drainage network. And calcium-magnesium ratios are very important in mineral soils, but in organic soils, and certainly in the soils that we're talking about in market gardening situations that are typically highly organic, these calcium-magnesium ratios become much less important because our soil structure becomes dominated by organic matter. The proportions of calcium and magnesium to do that, to support the integrity of the pore network, becomes secondary to organic matter because the organic matter is really doing most of the heavy lifting. These calcium magnesium ratios are still important. It's still important to have calcium dominance, but the whole idea of having ratios of 3 to 1 up to 7 to 1 as critical become much less important. Excuse me. So this is what our kind of standard soil test looks like. Left-hand side, upper level here, phosphorus, nitrate, ammonium, sulfur. That's kind of telling us what our macro elements are. We've got potassium down here too. And that's giving us information about the nutrient status of the soil. But where we see the calcium, magnesium, potassium, sodium, etc., given as a percentage of the soil, this is giving us information about how well that soil is likely to be structured. and its calcium magnesium ratio, again, how it's likely to be structured. Now, these data, these figures, the indicative guidelines, have all been developed for mineral soils. So like I say, when we're dealing with a highly organic soil, these become much less important. So striving to achieve a calcium magnesium ratio of 6.5 to 1 would probably end up wasting you a whole lot of money, because you've already spent a fair bit on compost, most likely. So I just was thank you to both Stan and Mitch for sharing some of their soil test results, and I've just picked a couple here. So this is Stan's top paddock, one of his productive paddocks, and I just thought I'd very quickly run through this to give you an idea of what I look for in a soil test and how I interpret results. And the nice thing about both of these soil test results we're going to look at is that they have optimal ranges, they have indicative values, which helps with our with lay interpretation of the soil test result. So up the top here, we've got N check. So they've separated out the nitrogen results from the rest of it. Again, because typically of our interest in nitrogen as a driver of production. So NO3N, that's nitrate nitrogen, NH4N is ammonium nitrogen. And we can see here that both of these are a bit on the low side. So typically we'd want both of these to be at least at 25 parts per million, 25 kilos per hectare, 8.3 and 3.4, giving us a combined 11.7. It says total available nitrogen here, but generally that nitrate and ammonium, they're both going to be available. But what it's telling us is that we have a bit of a nitrogen problem here in terms of how well production is going to be driven. And like I say, when we set the soil up for success, nitrogen becomes the accelerator. It determines how fast things move. But we want to set the system up. We want to optimize the soil for production so that when we do press the accelerator, we're using the minimal amount of fuel to get us traveling at the speed we want to go at. The pH in the soil is the next thing we look at. It's 7.13, slightly alkaline. That's generally fine. The sweet spot in soils is about 6.8, 6.5 in water. This is a reading in calcium chloride. So 6.5, say, in water is a sweet spot. But again, in a highly organic soil, we can drop our pH by a full 1 point. So where we have a sweet spot of 6.5 in a mineral soil, a pH of 5.5, 5.8 in a highly organic soil is entirely acceptable. The EC is a measure of salinity, and here this is perfectly fine. The total carbon is almost 5 here, and this equates to an organic matter reading of 8.6. I note that this lab is reporting organic matter of 7.7. I'm not quite sure what metric they're using, but a generally accepted metric is about 58%. 58% of the organic matter is organic carbon. It could be because this is total carbon, they're including inorganic carbon, which might slightly elevate this figure here. And the inorganic carbon will be things like carbonates and stuff. And if this paddock has been limed, as I suspect it has, there will be some inorganic carbon in here. The carbon to nitrogen ratio of 14.3, that's not too bad. The real sweet spot with carbon to nitrogen, as I said, is 12 to 1, and below 12 to 1, we're tipping that in the favor of microbial decomposition. So when we add nitrogen, we're tipping that ratio below this point. So if we were to add some canola meal, like Stan was talking about, that's going to bring this carbon to nitrogen ratio down below this number 14, probably below 12, and he's going to see a fertilizing response as a result. What we often see in highly organic and composted soils are these huge phosphorus results. Now, the guide here is 40 to 90, which is probably appropriate for a market garden soil. But as you can see, we've got 3 times more phosphorus than we require. The issue with this is that very high levels of nutrients can have antagonistic effects towards other elements. And in this case, very high phosphorus can have antagonistic effects on things like boron and other anions in the soil for plant uptake. And so in the midst of plenty, we can have an apparent nutrient deficiency. Potassium, on the other hand, is just a little bit low at 212. Again, in a market garden situation, we'd want to see that at a fairly healthy level of probably around 300 parts per million. That does depend a bit on the cation exchange capacity of the soil, and this one came in at 20.1, which is a high cation exchange capacity. It means it's got high nutrient holding capacity. The cation balance here: calcium 79%, potassium 2.5%, magnesium 70%. They're all kind of pretty good. We want to see the potassium level lifted from that, as I said, but sodium is good. And calcium magnesium ratio is 4.5. I wouldn't describe this as a highly organic soil, so this could I'm probably not going to play around with that too much, actually. My energy here is going to be more on trying to lift this organic matter level to sustain more organic nitrogen and better production from that point of view. So very quickly, these are Mitch's results. This is a very high organic matter soil. So at organic matter level of about 17%, we can begin to think about this as an organic soil. As you can see, most of the nitrogen here is in the nitrate form. We like to have nitrate in both forms. The plants require less internal energy to process nitrogen in the ammonium form, but if they get it all in the nitrate form, that's not such a drama. Sulfur is very good. Organic matter, very good. Cation exchange capacity, 32, is largely driven. There'll be a reasonable clay content in this soil, but having a very high organic matter content like this really increases the soil's nutrient holding capacity. So we'll get a big number here. This red, I think, means that this is too low. I would certainly not agree here at all. 68 is very much a sweet spot here, even if our magnesium is a little bit high, which only gives us a 3-to-1 calcium magnesium ratio. The fact, like I said, that we've got such high organic matter here kind of negates these numbers here, and I wouldn't be trying to elevate this calcium number above that. Manganese is a bit low, and I think it was Stan that mentioned that his garlic responded to a foliar application of manganese, which is a really good test. And a very easy thing to do is to apply manganese as a foliar and see if you get a response. And if you do, it is certainly an indicator that plants are not accessing, don't have sufficient access to that particular element. So we're talking about regaining the fertility of our farm. We want to learn how to take soil samples and read a soil test because we need to understand what we're managing. Read useful stuff. This is a shameful plug for some work that I did, but it's relevant to Tasmania, so I'm sharing it with you guys, and it's available through the Tassie government's website, and that's all about managing soil biology on Tasmanian farms. And we want to use the principles of regenerative agriculture to guide decision making. So if we're thinking about, should I do A or B here, reflecting back on the principles, it can be a very useful aid to help determine which way we should go. Whatever way we go, organic matter is the key. Composts ain't composts. Some are very young, some are old. Know what you're buying. Too much of a good thing is too much. Soil tests to know what you're dealing with. Compiled soil tests with observations, because healthy soils really do make healthy plants. There you go, the end.
Awesome. Thank you, Declan. That was really, really cool, and some very, very interesting information you shared. We do have one question from Luke, which is for Mitch. So Mitch, are you still there?
Yes, I'm still here.
Awesome. So Luke asked a question and he said, what depth are you going on your soil test? And are you looking at total nutrient pools in the soil or just plant available nutrients?
For sure. Yeah, thanks for the question, Luke. So yeah, 100 mils has been the standard as the minimum. I'm sure if anyone has taken samples before, cores are not always perfect, so it's hard to be super precise in obviously depending how wet or dry your soils are and obviously the type of your soil. So about 100 mils minimum, and that's generally what we've aimed for with all our tests, just to keep it standardized, of course. And we're looking at both, so we get total nutrient pools and available nutrients.
Awesome. Thank you, Mitch. We don't have any other questions at the moment, but I do have a question. So, Declan, you were going through the 3 different pools of soil organic carbon. And how do we know if our soil has different ratios? Oh, you're muted.
Don't you hate that? The only way we know is through testing. There are relatively cheap tests for particulate organic carbon, more expensive tests for humus carbon, and more expensive tests again for recalcitrant carbon. But in most market garden situations, it's unlikely you've got too much recalcitrant carbon unless you've been adding biochar. The landscapes that tend to have most recalcitrant carbon are ones with a history of fire stick farming. So most market gardens are going to have their organic matter in the form of particulate, labile carbon, and humus carbon. So the labile carbon is called a potassium permanganate test. It's relatively cheap, and that will just tell you what that component is. So the balance is most likely to be humus carbon.
Awesome. Thank you.
You're not going to be able to tell particularly by looking at the soil.
Okay. Yeah, right. Stan and Mitch, do you have any questions to Declan, or Declan, do you have any questions to Stan and Mitch?
I've got a question for Declan. Thanks, Declan, for analysing our soil test. So our soil, I may not have mentioned, our soil, is a relatively light, silty, with a bit of sand loam. So, we aim to try to build our organic matter as you suggested that we should. And we do that with green manuring or compost addition. Is there any recommendation? Should we just be adding more compost or doing more green manuring, or, we leave all the roots in? What's the best way for us to increase our organic matter? And the second part of that is, with adding canola meal as a nitrogen source, which presumably adds carbon as well. Is there any other alternative that you would recommend for us to add as a nitrogen source?
Look, I think canola meal is a good option, Stan, simply because it's got a fairly high nitrogen content. It's about 6% or thereabouts. And it's got a carbon to nitrogen ratio from memory of around 8:1. So it's got some carbon in there, as you say, but primarily its role is in bringing your soil's carbon to nitrogen ratio down so that nitrogen gets liberated for plant uptake, or at least it becomes super available to microbes who will then cycle that nitrogen and make it available to plants. So just looking at the top paddock, which was the example that I shared, the high zinc result that you have is most likely due to the fact that you've been buying in compost, and much of that compost, well, some of that compost may have come either from biosolids or from municipal sources. So us humans, when we're eventually extinct and the aliens come and do some fossicking around, and they'll find all these areas with high zinc, well, that's where the cities were, because we leave a big zinc footprint behind us humans, because we use zinc in a lot of things. Galvanized pipes and all that is just one example. So the zinc nearly always finds its way into compost and into soils. A zinc at that level, though, is not problematic. It's not going to concern us too much. As I said when I was going through it, I wouldn't be, I'm not going to try and particularly bring that pH down. You'll find with organic matter, with increasing organic matter inputs, the organic acids will slowly bring that pH down to around 7 or just under 7. What level of organic matter are you adding at the moment, Stan?
So in the top block, which is the data you were looking at, that's our deep compost beds. And so for the sampling that we did, we actually scraped all the compost away, and we were looking at the soil underneath the compost.
Yeah,
so I haven't really been adding much compost to that because there's still quite a lot of compost on the surface. I haven't tried to incorporate that into the soil beneath. In the middle block, we add just a small amount of compost, like a few millimeters every time we're doing a planting. So I'm not sure what that would equate to in mass per unit area, but I guess potentially we should be adding a bit more at that time. Is that what you might suggest?
Well, it depends a bit on how long you've been adding it. What's the answer to that?
Well, so in the middle block, it's probably been added for, what do you reckon, the last 7 or 8 years, and the top block since 2010, so 14 years.
So have you been adding the deep compost for 14 years?
No, in the top block, the deep compost has only been there for 4 years. And so before that, our management was more like what we use in the middle block, which is just adding a small amount. Yeah.
And sorry, just to clarify, did you put the deep compost on once in 2010, or have you been topping that up every year?
Once in 2020.
Sorry, 2020. Yeah. Okay.
Yes, once. Yes, at about 100 to 150 mil thick.
Yeah. Okay. So again, you know, the high. If you say that your soil is a light soil and has got a high proportion of silt in it, it suggests that it hasn't got a high clay content. So your CEC of 20 centimoles is relatively high. So most of that is probably being driven by mature compost, and that I guess aligns with what you have mature organic matter, and that aligns with what you've been saying, that you've been putting it on routinely for 7 years, and you've given it this big hit in 2020. I don't know that. I think putting on a big quantity like that is probably okay. Well, it's a good idea if we want to lift our soil organic matter. And having had 7 years of a small compost application annually certainly has bred up, a certain level of microbial population and activity in that soil. But still, putting a very large quantity on like that, you probably overwhelmed the capacity of that population to deal with what is, I guess, some orders of magnitude more compost than they've been used to processing. So that is probably equilibrating now because it's been 4 years since you put on that high quantity. And I would expect that that's something now that you just want to start a maintenance level of compost application. And that might still be 10 tons of compost a year, depending on the quality of the compost and its nutrient status, and particularly its carbon and nitrogen status. Because 10 tons of compost a year would equate to millimeters over the surface of the soil.
Yeah, that's right. Yeah.
Thanks for your question, Stan, to Declan. Declan, a question for Stan from Ben. What cover crop mixes have you been using?
Okay, yeah, that's we have done a few different things. So we have used a mix of legumes which include tick bean, which is the small broad bean, lupin, pea, and then some cereals, rye and oats. And then more recently, we've also been using Persian clover, Shafdal clover. So just depending on when in the year we cover cropping a bed, we use and actually like edible broad beans as well, that we use them as cover crop. So it just depends on when we're sowing a cover crop as to what we'll plant. The Persian clover has been great. It grows really well and produces a lot of biomass, and it breaks down pretty quickly. So I guess it's in that particulate carbon kind of group. So it works well when we chop it up and put a tarp on it, or sometimes we just don't even chop it up that much and just leave it on the surface, and it decomposes really quite quickly. Yeah, I think that's it. That they're the different green manure cover crops that we use.
Awesome. Thank you, Stan. And thanks, Ben, for the question. We are going over time a little bit, so we'll have this question as probably one of our last, and then we'll wrap it up very soon. So probably a question for Declan. And thank you, Beck, for asking the question. The question is there a place for tillage in highly compacted, laterite heavy paddock in Western Australia to regenerate the land to move towards it becoming an area for food production?
I would say the short answer to that question is yes, definitely.
Okay.
You know, I'm a firm believer in needing to break an egg to make a cake, as the old saying goes. And you know, the reason that we're resistant, the reason we're saying be cautious about tilling land and disturbing soil, is because of that. It triggers a release of carbon. So we tend to lose carbon in those situations. But if we're tilling the soil to put carbon in, or to put lime in, to improve the conditions in that soil, and then our management regime after that initial disturbance aims to complement that disturbance with cover crops, with you know, compost applications, with, you know, an improved management regime, certainly that disturbance, that tillage that breaks up compaction, that allows more air in, that allows more organic matter in, that improves conditions for plant growth, it's a no-brainer in that context.
Wow, there you go. Thank you, Declan. That's a great answer. There's no more questions coming through at the moment, so I'm going to pop in the chat box our link to the webinar feedback. So I'll give you guys just 30 seconds or so to grab that link before we close the webinar. And it's a simple 10 questions to just let us know what you thought of the webinar, and any feedback would be really helpful because this will guide us for future webinars and for future events to make them even better. So thank you everyone for coming along, and I hope you enjoyed this webinar today. I'll just have a couple more moments so you can grab that and copy the URL. And any final comments from Mitch or Stan or Declan before we finish up?
Yeah, I've got a bazillion questions I could harass Declan and Stan about, but I know we're running over time. So maybe we can continue the conversation another time. But yeah, thanks so much for the invitation, and I certainly really enjoyed hearing both Declan and Stan talk and being a part of it.
Yeah, from my point of view, I loved hearing Stan and Mitch's stories. I'm always in awe of you guys and the job that you're doing. It's like I'm totally preaching to the converted, so apologies if I'm overstating anything, but you know, you guys have obviously got the message well and truly, and it sounds like you're doing a brilliant job. Thank you, and thanks for organising it, Jacinta.
That's okay. Thank you, Mitch. Thank you, Stan. And thank you, Declan, so much for being part of this webinar. It means a lot to hear about your knowledge and learn from your experiences and understand like what are the next steps that you're taking for your different businesses. And all the knowledge that you've been sharing, Declan. Thank you.
Thanks a lot, Jacinta.